Okay, hi guys. So this is your first recorded lecture. We're going to start in on chapter 1, and so this is the introduction to genetics. So we're going to talk about some of the things that I'll expect you to know in this first chapter, and we'll talk through some of the concepts. Okay, so the objectives for this chapter. At the beginning of each chapter, I'm going to give you some objectives, and you can use these to help study. So by looking at these objectives, you can determine what you'll need to know for a test. So if I tell you to be able to differentiate between genetic disciplines, that means that you'll have to be able to know, if I give you an example of a study, for instance, that's been done, you have to say, okay, well, that actually falls under molecular genetics, or that's population genetics. So you'll have to know, you have to be able to differentiate between those two. You have to be able to describe the importance of genetic studies. So there's lots of reasons why we study genetics. You'll have to describe how model organisms aid in the study of genetics, and you'll have to be able to recall all the fundamental concepts that start on page 11. So a lot of the activities that we do for this chapter will be directly related to these objectives. So your activities help you to meet these objectives, and again, you can use these objectives to study for the test. Okay, so genetics is a branch of biology concerned with the study of heredity and variation. And that'll make a little bit more sense when I talk about the different disciplines in genetics. So one of the disciplines, so this is the first discipline of genetics. So this is also called classical genetics, but we call it transmission genetics now. And it's the study of how different traits are transmitted through generations. So this is the movement of traits from parents to offspring, and then when those offspring breed again to their offspring. So it's studying how the genes are actually affecting the physical appearance of offspring. The next discipline is molecular genetics. So this is the study of the structure and function of genes and gene expression. And we'll talk more about what gene expression means. So today, because of the advent of new technologies, all genetic disciplines are now molecular. So if someone studies transmission genetics, they're also using molecular techniques. And the third discipline we're going to talk about is population genetics. So population genetics is the study of variation. So they're looking at how genes vary within a population as well as between populations. So this is a map of human populations. And so, for instance, if we were interested in this big purple circle here, we would want to maybe know how much variation is found within that population that produces all different kinds of physical appearances, and then maybe we want to compare this purple circle with this orange circle. So what's the difference between these two populations? How do their genomes vary? So that would be population genetics. And then there's sub-disciplines within all of these disciplines. So for instance, within population genetics, we have conservation genetics. And in conservation genetics, we apply these same principles, but we're applying them to species of conservation concern, so endangered or threatened species. Okay, expression. So this brings us to the central dogma of molecular genetics. So you've probably heard this before, but this is basically expression of a gene. And so we have our DNA. It's transcribed into RNA. The RNA is translated into proteins, and this whole process is called expression. So we're going to talk a lot about expression this semester. So prior to modern genetics, there were quite a few theories about how heredity worked. This is just one of the examples, so you need to look in your book and make sure you understand these different types of theories that they had before. And so this is one. So this was the concept of a homunculus, and this was the idea that there was an actual fully formed miniature adult inside a sperm cell. of course, this was shown not to be true, starting out with Mendel's work, so we'll talk about him. So the theory that is now the basis of heredity is called epigeneticus. And so this is the idea that an organism develops from a fertilized embryo. So we have the egg and the sperm coming together to make a fertilized embryo, and then there's this succession of developmental events that are going to be occurring. And during those developmental events, genes are turned on and turned off to cause different stages to occur, and then there's this transformation of the embryo into the adult form. Okay, so the father of genetics is Gregor Mendel. He was an Augustinian monk, and we don't think of monks today as scientists, but back then, monks quite often did a lot of science, so they had the time and the means to be able to do research experiments. And so he studied both physics and botany, and he ran experiments in his monastery garden. So he tried to work with mice. That didn't go over so well with his fellow monks, because mice stink and they're hard to keep alive in comparison to a garden. And so he started working with pea plants, and over the course of 8 years, he ran experiments on over 10,000 plants. So this is a really rigorous, strong experiment that he performed. So again, he worked with pea plants, and the reason that he did is because of their very clear physical traits. And so on the left, you can see there are pink flowers, and there are or red flowers, depending on what you want to call that, and there are white flowers. There's different heights to the plants. They just have 7 really easy identifiable traits that he could work with. And so what he did was he actually fertilized specific plants. So he would go in and cut off the anthers of the plant so that it could not, so that fertilization couldn't happen unless he actually performed it. And so then he would dust the stigma of the plant with pollen taken from a specific father plant. And then he would put a bag over it to make sure that it didn't get fertilized by any other pollen. And so this is some of the first records of selective breeding. So this is the same thing that we do. Some people do with pigeons, we do this with dogs. So this was, he used selective breeding to perform his experiment. So one of the things that he found not to be true was blended inheritance. So at the time, everybody thought that a red flower and a white flower, if they were bred together, they would produce a pink flower. But his work proved that that's not what happens. And we'll talk more about that in chapter 3. So remember, too, that at the time, Mendel didn't know about chromosomes. He didn't know much about cell division or the structure of DNA. So a lot of the things that we know now, he didn't know. So he was just doing this transmission genetics where he would breed certain plants together and then look at the offspring traits to determine how traits were being passed on. Today, we have more technology. We have model organisms. So some of the common model organisms are mice and fruit flies. And the goal with a model organism is to have an organism that you can easily control. So they need to be easy to rear, so easy to feed and keep alive. They need to be easy, reproduce easily, so they need to reproduce in captivity. And the faster the generation time, the easier it is to look at inheritance. So fruit flies, for instance, you can have multiple generations within a couple of days. And so, so that's a really nice aspect of a model organism. They're also based on similarity to a process or an organism. So new model organisms are being developed all the time, trying to find ways to study different human diseases. So we need a system that's very similar to humans if we want to understand how a medicine works, for instance, or a treatment. We also have plant model organisms, so on the left is corn, and on the right is Arabidopsis. These are all model organisms as well, so not humans, but mice, chickens, Xenopus is a frog model organism, zebrafish, Drosophila, and then a nematode, which you've probably heard of as C. elegans. So again, with the advent of technology, we're developing more and more model organisms. The sea urchin is now considered a model organism. And here at Nichols, they're doing some work in collaboration with some other universities to develop spotted gar into a model organism. And so, again, it's just they're trying to develop more and more organisms all the time so that they have what they need to have a very similar process to look at human disease. We are now in what's called the omics era. And so this means that instead of studying genes, we're studying genomes. So we're studying whole genomes, all of the genes that make up the genome. And so this is due to new sequencing technologies as well as bioinformatics, both of which you'll become familiar with during this semester. And so if you study all of the DNA, you're studying genomics. If you study all of the RNA, you're studying transcriptomics. And then all of the protein is proteomics. There's also the study of metabolites, and that's metabolomics. So there's lots of ways that we've just expanded what we're learning to these massive data sets to really get more and more information. And so it's really exciting to be a part of genetics right now because of all of the new things that we're doing. Okay, so that is it for today, or for this presentation. And then we'll have some activities that we'll also do that will be a part of this chapter. Okay, bye-bye.